This session is about TVB processing pipelines with knowledge graph annotated outputs. What is the goal of this processing pipeline? In a nutshell, someone has MRI data and wants to study brain networks. So the input of this pipeline is MRI data, and the output are a specification of structural and functional brain networks, basically. So this is the one sentence answer. The more detailed answer is a structural connectome is basically an aggregated description of the brain's white matter fiber infrastructure. So we have a full brain tractography and we have a parcellation of the brain. And then we basically aggregate the fiber tractography result, which was done in diffusion MRI data. And what we get out of this is a matrix, a weights matrix, which specifies the coupling strengths, the interaction strengths between brain regions, between each pair of brain regions. And a matrix which specifies the length of, or the average length of fiber tracks between brain regions, which can later be used to estimate the conduction delay, the delay of interaction between brain regions. As an input for the pipeline, we need T1 weighted MRI data. We need diffusion weighted MRI data. So these are necessary for constructing a structural connectome and for constructing the brain parcellation. We can also use T2 weighted MRI data to improve the registration with fMRI. fMRI data we use to construct functional connectomes, which are often used either studied alone empirically or as a fitting target for optimizing the parameters of a brain network model. And pipeline also outputs or it processes also EEG or MEG data and outputs source imaged EEG or MEG data. So projects this activity into the source space at different cortical locations. So here is now an overview over the different outputs of the pipeline. I think you cannot see it already. Hopefully it comes in a second. Anyway, no, there it is. So we have structural connectomes, which are basically just two matrices, as we just saw. We have region-wise fMRI time series. So this is basically the average fMRI activity over all voxels in each brain region. From such region-wise fMRI time series, we compute functional connectomes, the functional equivalent of a structural network, so to say. The pipeline also outputs surfaces, so we have triangulations of the cortical surface, of the inner and the outer skull surface, and of the scalp surface. We have lead field matrices, which is an aggregated representation that associates the source space with the channel space. So if we simulate activity on a source space, for example, local field potentials, we can use such a lead field matrix to project this activity into the EEG or MEG channel space and simulate EEG or MEG. And we also output brain parcellations, which could, for example, be used as masks for further analysis. What is the architecture of this pipeline? So the TVB processing pipeline at eBrains is basically a workflow of 3 Docker containers that are run one after the other. The first one is called TVB Pipeline SC, which is basically a clone of the BITS app and Matrix 3 connectome, which does the structural processing. Then we have a clone of fMRI Prep, a popular software for fMRI preprocessing. And then we have the TVB pipeline converter, which takes the outputs of the first 2 containers and performs some further processing on it, and then generates the outputs in bits format and in TVB ready format. And this workflow uses several services from the eBrains cloud. As a front end for this workflow, we have a Jupyter notebook in Lab, which is eBrains service to serve Jupyter notebooks. So it's a Jupyter Lab client, basically, where each user has their own private space, basically, where they can run notebooks. Then Drive is eBrain's space to share files, and there users can upload the input data for the pipeline and download the results and transfer them back and forth between the backend supercomputer, which are used to do the pipeline processing. And then we have the wiki, where documentation is, and where users may work collaboratively on documents in order to make research with such workflows. And we have the supercomputing backend, which is in this case Disdain from the CSCS facility in Switzerland. As Peter already showed, these eBrains collaboratories are accessible via this wiki.ebrains.eu URL. And when you search for TVB, you will find all the TVB services. And basically, this TVB pipeline is then the link to the Colab, where the notebook is linked, and also a video tutorial. And associated with this collaboratories, a Drive space where the notebook is hosted and where example data is, and which basically needs to be cloned by the user in order to edit and manipulate it and change it. As I mentioned, the front end of the pipeline is Jupyter Notebook, which has documentation and code snippets interspersed. So there's always some explanatory parts which explain the steps, and then there are code snippets that perform these steps. And basically what is done in this Jupyter Notebook is it explains how a user can upload their bits data set to the eBrains Colab, how they would access data in a Jupyter Note, how they access data in eBrains Drive from an eBrains Jupyter Notebook, how they upload from the Jupyter Notebook the data to the supercomputer via PyUnicore, which is the interface which links all the supercomputing facilities, basically. Then we create and execute Slurm batch job scripts to execute jobs on a supercomputer that executes a pipeline. And then we show how we download the results and how we finally download them to the computer, to the local file system. So basically all the user has to do with such a notebook, the notebook basically does everything more or less automatically. The user has not to specify too much, so just a few parameters like what is the name of the data set, in which folder is it located, what is the name of the collab, and so on. This is at this stage all the user has to specify. Later they also have to specify a few features of the data set. And basically, then this Jupyter Notebook goes step by step through all the necessary steps, runs the 3 containers one after the other, shows the format of Slurm job scripts for the job manager of the supercomputer, and how we can provide command line arguments to the Docker containers in order to configure the workflows. What are the data formats of this pipeline? So I see the screen is lagging quite a few slides behind. I will wait a little bit. So now I have a slide here where I see input in bits format. So this is the input to the pipeline. What we need is diffusion weighted MRI. We need functional MRI, anatomically T1 weighted MRI. We need field inhomogeneity maps in order to correct susceptibility-induced distortions, AP distortions of the data. We need for diffusion susceptibility distortions also pairs of phase encoding reversed B0 and ideally also gradient acquisitions in order to perform such in-field inhomogeneity corrections. As we can see with this folder tree here, this is typical bits folder schema. We have the metadata files like dataset description, JSON, we have the participants, TSV, a README, and a changes file, and then we have this typical folder schema with bits, subject as key value pairs, basically. So in order to, for example, specify the subject, to specify the session, to specify the type of data, anatomical, diffusion, or functional, and then we have the data files accompanied by JSON sidecar files that describe the metadata of the data files. The outputs of this pipeline come in two formats, once in TVB-ready format and once in bits format. So what are the outputs? We have, so basically the output format for the TVB are basically plain text ASCII files where we store matrices or column vectors, and as a separator we use white spaces and new lines, what we store there in these output files are the weights. So this is an n times n matrix of coupling weights. We have the tract lengths, we have the centers of brain regions, we have the surface areas of the cortical area of each brain region. We denote whether it's cortical or subcortical, have an estimation of the average orientation of this area, whether it belongs to the left or right hemisphere. Then we have all these surfaces outputted as triangulation. So we have vertices, triangles, and normals. Then we have a region mapping that associates each surface vertex with the large-scale brain region where it is located. We output the locations of the sensors, which was found by taking the reconstructed scalp surface and projecting a standard set of EEG electrodes or MEG electrodes around the surface or on the surface. And then we have a projection matrix or lead field matrix where we basically associate source space and channel space. And basically more or less the same data now in bits format. So here we have, this is a bits format specification for derivatives data. And there we have typical bits features like key value pairs in file names that describe the features of this data. What is the API of this pipeline? I basically already mentioned it. The interface with which users interact with this pipeline is basically command line arguments that we provide to Docker invocations, basically. Here we have just a selection of typical command line arguments that we would provide for the structural preprocessing, for example, the choice of the parcellation that we want to use, or the number of streamlines that we want to provide, or the analysis level, whether this is a single subject or whether we again run this pipeline then over a group of subjects. fMRI prep, the fMRI preprocessing container allows more options for configuration. So they have a very rich interface, which allows a lot of, specify a lot of command line arguments basically to configure the workflow. The TVB pipeline converter takes all its required inputs as command line arguments. It's not very much, and the user doesn't have to care basically about setting these arguments. The Jupyter notebook basically cares automatically about this. Okay, a few words about software maturity, integration, testing, versioning, and deployment. Software maturity. This is a research software with a very complex workflow. So especially these structural connectome workflows, there is like no black box or out of the box solution where you just dump in your data and hit a button, and then at the end comes a plausible output. So such pipelines typically work for the test data set, which were used to set up the pipeline. But if you then use other data sets, they often break down. So when it comes to software maturity, one has to say that such pipelines often need interventions. So they often users have to even dig into the code and change code at some places in order to make such pipelines work. But this is all fully documented. This is all open source. It can all be accessed and manipulated. And there's also a lot of support from various directions. The knowledge graph integration is not fully implemented yet. This is a quick and dirty solution yet. I will provide details what this means in a minute. Integration, the software is tightly integrated with the other core eBrains services like Drive, Lab, the supercomputing backends, and the wiki. Testing, as I mentioned, the pipeline runs successful with test data, but it's likely that features of data sets, for example, data quality, or also, for example, features of the subjects or patients make the pipeline break down. Versioning is achieved by giving the Docker images and GitHub repositories version numbers, and by keeping these older versions also online, and by reporting the container and the version number, a workflow should be exactly be reproduced. So in order to reproduce something from a publication, a workflow from a publication, all that is necessary is to specify the container and the version, and then this workflow can be reproduced. Deployed, as we saw, it's in the eBrains cloud and on Pistain. Now a few words on knowledge graph integration. So knowledge graph is the basically a database that stores neuroscientific data and annotates this data with a lot of metadata. And for that, the MINDS format is used. And when we started this work, the MINDS format was basically an Excel file, basically, where we have different key value pairs for different metadata entities. And basically what we provided until now is basically an automatic way to fill up such these different fields. But the better way, which will come soon, is the new Open Minds format, which is closely before being released. So this is a really exhaustive schema for experimental data, computational models, and software. And this is based on JSON files, and these JSON files are linked by JSON LD schemas. Basically, the Open Minds team will then provide generators and validators that basically transform, for example, or automatically read out bits metadata and transform it into Minds format, for example. So this is the Open Minds GitHub repository. Will come in a second. And just to give an overview how such a JSON metadata schema will look like then. We haven't arrived at the slide yet. Can already go a bit into future here. Wait a second until it refreshes. Okay, so this is the folder tree, basically. We have, for each possible entity, one could think of basically a JSON file. So the main entity of the metadata is basically the dataset JSON, which, for example, specifies contributors. These contributors are then persons that are stored here in the subfolder person. And these persons oh no, sorry, I'm at activities right now. So for example, you can also have activities here, and basically it's a linked schema. So we have for each element of this metadata schema, we have one file, and using these JSON LD path schemas, we can basically link all these files together so that we have a network of metadata basically where we can describe all the different features of the data set, like what species the data set is, what age, and so on. And yeah, so this will come as soon as the Open Minds schema is released. A few words about computing requirements. So what makes this workflow expensive is mostly the tractography step. So all other steps are relatively cheap and could typically be run with relatively standard resources, which also can be found in a normal home computer. But especially the tractography step takes a lot of CPU time. So here we would run it for on 36 cores for 20 hours. And we'll end up for one data set with around 833 core hours to compute one subject fully and compute the entire structural and functional connectomes of the subject. Then finally, a few words about security requirements. So the eBRAINS cloud is, it's a cloud. So it's accessible via the internet. So it's connected via open networks basically, and it works with shared resources. So this is basically makes up a lot of vulnerabilities, opens up a lot of vulnerabilities that could occur at many different points here. So since we are sending data back and forth via the internet, there's always the possibility that data is intercepted in transit through these open networks. And even if data is encrypted, loss of cryptographic keys could then still lead to data corruption. So in general, cloud infrastructures always typically suffer from the same vulnerabilities. So because the cloud is internet accessible and also via remote access, we have via web browser, basically, we have a lot of vulnerabilities also on the side of the web browser, on the side of the client, that make such an architecture inherently vulnerable to cyber attacks. Which can lead basically to isolation failures of all sorts, where basically somehow some other user of this shared resource, which is a supercomputer or also the supercomputers on which the front ends run, vulnerable to being breached basically, and to enter private user spaces. And since this hardware that's somewhere in some facilities, there's also, of course, the problem that this hardware, someone must guarantee that this hardware is protected and that data after they have been used, the data is securely deleted, basically. So even if you hit delete, most operating systems won't just delete something, they just clear a logical flag. But to really so even if you overwrite something on a magnetic hard drive, then you still can recover this information if it was overwritten like 8 times or so, with relatively simple technology actually. So this is of course all. And with this, I come to the end of this session.